Hi, thank you so much for the invitation to be part of your class. I want to talk to you about microbialites. This is a model study that we've been working at the Bacterial Ecology Lab, Mexico's National University. Microbialites are structures made by microbes. So, through their different metabolisms, there's going to be precipitation of carbonates, and these small rock like structures form bacterial reefs in different aquatic ecosystems. If we take a closer look at their composition, we're going to find a wide range of bacteria, archaea, and viruses mainly, that are basically cyanobacteria and diatoms, purple and green sulfur bacteria, chloroflexi, loads of proteobacteria and planktomycetes, and archaea. And together, they're going to couple their metabolisms between night and day, and we're going to be able to see both oxygenic and anoxygenic photosynthesis, formation of biomass, and then respiratory pathways, mainly fermentations, and basically their cycling of carbon, nitrogen, oxygen, phosphorus, sulfur, making these self sustainable communities. In this microscopy image, we can see filaments of cyanobacteria and associated minerals to the base of those filaments. And I'm going to talk to you more about that. But basically, there are metabolisms like photosynthesis and reduction of sulfate that promote carbonate mineral precipitation. And then there are metabolisms such as fermentation, denitrification, that will promote dissolution of carbonate minerals. And so microbialites live in this equilibrium between precipitating minerals and dissolution of minerals, and this is how the structures are formed. Stromatolites in the fossil record constitute the oldest evidence of life on Earth. They have been dated in over 3.5 billion years. And stromatolite basically means stroma, layer, and lithos, rock. So these are layered rocks that occur in the fossil record. And microbialites are their modern counterparts, with microbes that form rocks. And we know from the geological record that way back in the Archean, when we have the oldest evidence of stromatolites, these were very abundant in different coastal and inland ecosystems. Their abundance rose towards the Proterozoic, and so there are several geologic locations, and the most ancient we know are in Western Australia, but also in North America. And then in the Cenozoic, there are modern Holocene microbialite reefs, and basically their evolution is associated to the oxygenation of our atmosphere. So you'll remember that the Archean atmosphere didn't have as much oxygen as we have right now. Actually, it was a reducing atmosphere. And a bubble at a time, these communities were liberating oxygen. And, little by little, there was enough oxygen towards 2,450 million years ago in the Great Oxidation Event, where we can see banded iron formations in the geological record. And we know that this oxygen comes from oxygenic photosynthesis, and we think that microbial mats and microbialites were the type of communities, plus all the planktonic cyanobacteria that were responsible for this oxygenation. There are ancient microbialites that are pre-cyanobacterial in the Archean, and then mid-Archean towards the Proterozoic, and then all the modern types of microbialites have cyanobacteria that are the main constituent in biomass. They are the largest microbes in the microbialites, and they make this biome possible through a series of interactions with all the other diversity of microbes that constitute these communities. Once the atmosphere of our planet had enough oxygen, that gave rise to eukarya. And eukaryotes started eating microbialites. And so today we find these bacterial reefs in aquatic ecosystems that are not that don't harbor as many eukaryotes. So, for example, we find them in ecosystems that have very low levels of nitrogen and phosphorus, which limits, for example, algae. And algae, if they are present, they will outcompete and grow over these microbial structures. We can also find these types of communities in sites that are extreme, for example, in their salinity or ionic composition of the water. And so there are different environments that will sustain these communities but are easily outcompeted by eukaryotes when environmental conditions change. So we know now that modern microbialites capture CO2 in carbonates but also in organic matter. They will be actively liberating and producing oxygen. They are sites of strong nitrogen fixation, cycling of phosphorus, sulfur, and these are self-sustainable communities that are highly productive in terms of carbon fixation. So we've been asking basic questions of community ecology as to who forms microbialites, where do they live, how do they live in these environments. And so our main theoretical background and concepts are, well, there must be a regional species pool of microbes that through some random filtering and abiotic conditions, plus all the biological interactions between the components of these communities, will lead to the local species assemblages. And we want to know if sites that harbor microbialites, say for example in Mexico, that's a country that has several sites with modern microbialite reefs, if microbialites in these sites have the same local community assemblage, or if they're all different, and to what environmental variables these are related. So we have worked in different environments, both in desert oases, in crater lakes, and also in coastal lagoons. We have a marine site in Cuba that we've included in our analyses. And we have used a different, array of techniques depending on what we want to answer. So for example, to understand how they are constructed and formed, we've used different forms of microscopy and imaging techniques. So for example, here I show you a stereo microscopy cross-section of a microbialite, where if we look at it from, above, we can see these dark filaments associated to minerals. And if we go, further in with electronic microscopy, we can see these filaments that are, they are part of the Nostocales, a group of cyanobacteria that are very big microbes, and they have heterocysts that are specialized cells for nitrogen fixation. They would be at the basis. You wouldn't be able to see them here because they're embedded within carbonate mineral matrix. And with Fourier transform infrared spectrum microscopy in collaboration with our colleagues at the LBNL, Hoying Holman and Patricia Valdespino, we can see that the carbonate precipitation is associated to these specialized cells, heterocysts, as well as the polyphosphate storage. And so we're starting to understand functionally how the communities are being formed. There's also work that suggests that carbonate mineral precipitation associated to cyanobacteria occurs in the S layer that's part of the outer membrane where we have EPS. And so photosynthesis is going to raise pH locally, and this will conduct the precipitation of carbonate minerals. And the type of carbonate mineral will depend on the environment. So we now know with the work in collaboration with Dr. Teresa P. Puig from the Geology Institute that microbialites from different environments have a differential mineral composition, with calcite being the most common, hydromagnesite as well, but we also have tiny reefs with basically aragonite in certain places. To understand who and how we have, the particular local assemblages of microbialites, what we've been doing is community composition data matrices, basically with different next-gen sequencing techniques. So, for example, we do total DNA extraction from, we design the sampling strategy to represent each local assemblage and then the geographical range of these assemblages. And then we do, for example, hypervariable V4 region amplification of the 16S rDNA. And doing this, we now know that there are microbialites from different parts, from different regions, will have basically the same phyla, where we have Proteobacteria, Planktomycetes, Cyanobacteria, Chloroflexia is the most abundant, a bit of Archaea, and doing ordination of this diversity, we know that there is biogeography. So microbialites that come from local assemblages that are geographically closer together will be more similar than to those that are farther apart. We have also compared their transcriptome to analyze, for example, the nitrogen cycle to try to understand if the same organisms are responsible for the transformations of nitrogen. And we know that there is a great diversity of microorganisms responsible for nitrogen fixation, oxidation of nitrite, and reduction of nitrate, reduction of nitrous oxide, and denitrification as a complete pathway. We also know that the expression, for example, of nitrogen fixation is among the most important in microbialites. And so we've gone ahead and measured with acetylene reduction the rates of nitrogen fixation for these communities. And we know that in general, heterocystis cyanobacteria that fix nitrogen during the day are the most important N2 fixers in these communities. This has been part of the work of Isla Beltran, Carla Centeno, and Rocio Alcántara. Doing environmental DNA shotgun metagenomics with Daniel Cerquera, what we know is that different microbialites fix nitrogen, do the pathways of the nitrogen cycle I just talked to you about, that carbon cycling for oxygenic photosynthesis is very important, but also other types of anaxigenic photosynthesis. For example, reverse DCA and the reverse acetyl coenzyme A pathway. Actually, if we add up the metabolic fluxes of all the anaxigenic photosynthesis pathways, they are as important as oxygenic photosynthesis. And this helps us understand that even though we're working with modern communities, the signal of pre-cyanobacterial microbialites is still part of these modern communities. A site that we have focused a lot of research on is Laguna Bacalar in the southern Yucatan Peninsula in Mexico. And this is because the largest freshwater microbialite reef exists in this aquatic environment. And so, what we have done is try to understand if we have the same microbialites along the lagoon or if they are different, if we have bioregions. And so, in this map, I show you some of the sampling sites in Bacalar Lagoon, and we have basically done the same. In each sampling site, we choose around 6 to 10 individual microbialites. We have subsamples for each of these, and then we construct a composition data set that we start to explore. And for example, we know that again, Proteobacteria, alpha, delta, gamma, Cyanobacteria chloroflexi, but also Firmicutes, Bacteroidetes, Verrucomicrobia, Planktomycetes are among the most important phylum in these structures. But when we do the ordination, we find that in this site we have two main bioregions. A bioregion located towards the north of the lagoon that has almost twice as much diversity than the second bioregion that's concentrated in the southern part of the lagoon that has, well, we don't know if it has lost diversity or it's simply less diverse than the first bioregion. Well, we have included analyses that incorporate the phylogenetic structure of the community as unifrac distance measures, which are really interesting because you don't only do the composition of the community, but you also add the phylogenetic information. So you have to make a phylogenetic reconstruction where you place the sequences that you obtain in the study. And there is information regarding how closely related each of your amplicon sequences are to construct a distance matrix. And so you can understand if different local assemblages share a same origin or if they come from different events, which is also important to understand the evolution of these communities. In the case of Bacalar, what we know is that the main environmental variables that explain the differences in composition are related to tourist intensity and environment quality. So, for example, in the northern part of the lagoon, where we have more diversity, there is low impact tourism, and the water quality is better because it has less concentration of reduced forms of nitrogen and available forms of phosphorus, versus the southern region of the lagoon where we have more intense tourism and we also have higher concentrations of nitrogen and phosphorus. So this has led us to propose sites for conservation of this microbialite reef and sites in the lagoon that can be used more, that can have like a stronger use for tourism, because this site is very, it's very important for the local population to have tourist activities, but you have to do this in a sustainable way. So understanding the patterns of diversity is fundamental. Associated to Bacalar Lagoon is a sinkhole called the Blue Hole, Cenote Azul, where we have studied microbialites that develop up to 30 meters in depth. But in our explorations, we know that this microbialite reef goes further deep. And here, what we have is a very interesting ordination of the composition with deep microbialites in 20 and 30 meters that are different to shallow microbialites that go from surface all the way down to 10 meters. And basically, the composition, even though it is similar, at the phylum level, we start to see, for example, something that we don't see in Bacalar Lagoon in the surface microbialites. Here in the blue sinkhole, what we have are new organisms that we need to continue studying, like a completely unknown family of cyanobacteria, several Thaumarchiota, and Planktomycetes that need more research. And so we're going to start doing, well, we're already analyzing some shotgun sequences to try to reconstruct the genomics of these microbes. Bernardo Aguila is doing his PhD studying the cyanobacteria and microbialites from Bacalar Lagoon, but also from the blue sinkhole. And something that we are proposing is that the deep microbialites that grow in this blue sinkhole are older and relate to the origins of the lagunar system, going way back to 13,000 years ago, whereas the modern surface microbialites are, under 5,000 years in age. And well, so this is our study model. And I think, I hope that now when you see microbialites, you'll remember that they're actively fixing nitrogen and producing oxygen, are really productive communities. And well, I hope that you have a lot of questions. And well, I really appreciate your time and attention. You can find us at the Bacterial Ecology Lab, and this is my email. So thank you so much for your time.